Hello.
Hello,
Hello, interwebs.
and welcome back to Philosophizing. Um,
Yeah. My name
yeah,
is
let me just
Ruby.
check. I'm Nathan.
Hope you can
Let
hear
me just
us.
check that everything is working okay on the audio. Um,
Great.
While we're doing that, how's your week been so far?
Oh, today was so hectic. I had to give a
Why?
lecture today.
Oh, is it the neurodegenerative disease one?
It is. It is. And if y'all are interested in listening to it, I gave the very similar talk last year around the same time. And it was
I
like,
believe I believe the talk is actually on our YouTube.
yeah, it is on our YouTube. But I had to like
It's
remember
pretty
all the things
interesting.
again.
Yeah.
It was really helpful, like some of the things that Nathan added, like he was showing us gels and like how to read stuff on during the talk. I incorporated it into the actual talk this year, which is I think helpful. They scored better on the quiz at the end of the talk, so I guess
Oh,
that's like
interesting. So, like, do you think that you like have smarter students this year? Do you think like you did the class better, or
that is such a good question because my N is 2. Oh, actually, the N is not 2. The N is 16 last year and 14 this year. So, but yes, there's only 2 samples, right? Like one last year and this year, and the N's are a little low, but they did demonstrably better this year. Um, Not that that's like, or maybe okay, maybe it's not on them. Maybe it's I performed better this year. Oh no, Nathan, I can't hear you anymore. You adjusted something and now you are muted. And you're texting me. Am I muted?
no?
Oh,
Is
oh,
it
you're
working
back.
now?
I can hear you now. Yeah, yeah.
Oh, weird. Um, Sometimes these headphones try to connect to two different like audio sources, which is weird.
Got it.
But yeah, what I was saying is like I feel like this is actually a good segue into our talk about reproducibility.
Yeah. I don't know
Um,
what
I
like
didn't
I
actually plan that, but
yeah, no, I don't actually know how I came up with this topic. I think I was in the kitchen somewhere and I was like, this is a kind of a problem and it's interesting and I think it'll be good for philosophizing. But I was just like, we must give it this week. I don't know why I decided that this is the
like why
week
you
we
were
must do it.
compelled to do it this week.
Yeah, I was like, man, I'm gonna do a whole lecture and also this deep dive, and I feel like I might
Was
not
it
be
like
able to.
was it like you were reading about these or working on these like disease things and then you were like, oh, this research doesn't seem very reliable, or
Okay, you know what? It might have been. It might have been. I read a paper for Journal Club and I got angry because I had tried to do this experiment and it didn't work, and yet they got published by someone
uh,
else, not me. Um, and then I was just sat there. No, I don't. It wasn't like a direct one to one like this caused it. It's just like this feeling that's always been
right.
there, and it's like how you have this like I don't know if you have it, but I have like a boogeyman, which is like any like I have this like compulsion to like publish things that are correct, and so like the boogeyman is someone else reading my paper and then doing the exact thing and then not being able to like, get the same result.
Like not finding the same result.
Yeah, that is my boogeyman because then I was just like, oh my god, that stresses me out. Like I genuinely, because like I don't know, like I think that's all we have as scientists, right? We have a reputation,
Yeah.
and if that happens enough times, like it gets degraded. And I think that is one of the core reasons that society in general has so little trust on science because 1 week we're like don't wear the masks and then like 2 months later and it was like never mind you wear the masks. Do you know what I mean? Like we're
Yeah.
um
Although I don't know, like I guess I'm not like I feel like that's not. I mean, maybe reproducibility is part of it, but I think that like there's other parts to that too. Like I mean, there's all the jokes about like, oh, do eggs cause or prevent cancer, and like
yeah.
how there's a bunch of studies that say they cause it and a bunch of studies that say they prevent it, and like I think that's like less about reproducibility and more about the fact that like just like whatever eggs are doing, they have a very small effect size. So like, and actually honestly I think that this is an interesting thing that I think is tangential to the reproducibility discussion, which is that like we often think about studies as like either they found a thing or they didn't find a thing, but in many ways like what you're really doing is you're saying kind of like you're estimating the effect size. So you're saying like, okay, how big of an effect does eating eggs have on cancer? And the effect could be 0. It could be positive, meaning it gives you cancer, or it could be negative, meaning it prevents cancer. And like like if you think about it as kind of a continuous thing, some of these things make more sense. So it's not just like yes or no. It's like
It's
there's
like
an
types
effect
of
size
data. It's like,
somewhere on
is it,
the spectrum.
does it fall
Yeah,
into buckets or does it fall into like a spectrum? And you should pick different statistical tests for that. And
right.
but
And
anyway, well, wait, can I do the
yeah,
introduction? Because I feel
sure.
like this will help
Mhm.
us like organize our thoughts. So basically, the idea of the reproducibility crisis is basically that. The idea that there's a surprising number of published studies that are difficult to reproduce or can't be reproduced. And so in I think right around the time I started grad school, like 2014, 2015 era, there was a bunch of papers that came out, and one in particular came from the Center for Open Science, and it estimated, so it was like a meta-analysis, and it was used to estimate the reproducibility in science. So they basically took all these papers, and they went in and did the experiments again. And they found that only about 36% of those were like psychological studies, or like psych and cog sci studies, could be reproduced. And then that was like, it was published in Science. And then like everyone freaked out because they're like, oh my gosh, like 64% of this stuff couldn't be reproduced. Like, oh my God, like this is a crisis. And there was a huge panic. And like this also coincided with I forget, this like Retraction Watch got this guy, and he was
Mhm.
just like making up a lot of data. Like it was this guy that was doing science about like food and correlations between like how much you eat when it's placed on the table versus if you have to take it out of the cabinet, like things like that, like really like
Oh, yeah, was that
freakonomic.
the ego depletion thing?
Oh, what was his name? It was like Brian something. I don't want to like slander some rando if I can, but I can find the links. But it was like the downfall of this guy, and it was just like all over the press, and it was a really bad look. So in that era, we were like, oh my gosh, and so it actually made a lot of scientists, and including this like open science field, be like, hey, like let's go and like test some of these things, and so then they tried to reproduce a bunch of cancer studies. And so for I guess like background, a lot of things that are proved or like has shown efficacy if rodents, when they translate into humans, a lot of times there's this thing called the valley of death, where like they take the clinical trials, like it goes to safety, and then like it's not, like doesn't do anything or there's like
Mhm.
low like not great results, and they're like, why is this happening? And so the cancer biology field was just like, yeah, let's do this. And so then this eLife paper came out, and it was like reproducibility in cancer bio. And they also found, like, so they went through like the most high impact papers, like 50 of them, and then they repeated experiments from those labs, like just bought the same reagents, like did
Mhm.
the experiments the exact same way, and that was really rough. So I think here's a bit. So let's see how many were able. Um, so the problem was like not all this experiments were explained and detailed enough, like in the methods for them to like do it perfectly. So like that's the caveat. But like uh I think they said that only like they could only do it like 41% of the way, the replication. Like they had trouble
Mm,
basically recapturing even the experiment itself, not let
right.
alone the
Like
results.
so like only 40% of them they were actually confident that they were able to replicate it.
Yeah, they didn't even like, they're like, we don't even know if we can do it. It wasn't even the results. Like we were just like, we're gonna take these big studies and trying to just redo the same experiment again. And so that's the big I guess like this is the boogeyman.
The crisis, yeah.
Yeah, this is
Yeah.
the crisis.
Mhm.
And I
Like
think
when people
the
talk about the reproducibility crisis, this is the crisis, is that like all the science might actually just like be wrong.
yeah, and and I really like this article that Dorothy Bishop did, and so she called she like kind of like put all the parts of the problem into like one analogy, and she called it the four horsemen of the reproducibility apocalypse. And so the first boogeyman number one was publication bias, which is we like to publish things when they have a huge result, and negative results are less likely to get published and less likely to be reported, less likely to be documented, less likely to be talked about. And so when you only talk about basically a really small portion of things that could work, and you bias it because that's what the big journals, right, like the big journals want splashy titles and like interesting things, paradigm shifting things. You end up biasing the results that are being documented. And so that's boogeyman number one. Boogeyman number two is like poor statistics, low statistical power, and like publishing stuff that are barely significant. And like that's the second one. Like it's really hard to sometimes it's really hard to do replicates, and scientists are shitty statisticians because we didn't study statistics explicitly, and we picked the wrong tests, and we just but it's just like not trained. The third one and fourth one are p hacking and harking, and those two kind of come in hand in hand. So like p hacking, so for me what p hacking in my field is if you collect, let's say, like 5 animals worth of data, and then you look at the data and it's like, oh, it looks like there's a trend. I'm just going to keep collecting animals until this trend becomes a value that is big enough to publish, which is usually canonically like 0.05. So like my p-val is greater than 0.5, I'm going to stop collecting now. And so that's p-hacking, right? So if you don't predetermine your sample sizes and like, do you know what I mean? That's p-hacking.
Yeah.
And harking is the equivalent of that. It's basically like if you explore the data after you collect the data. Does that make sense? Which sounds wrong, right? Like you're like, wait, a lot of people collect data and then they go fishing. Like that's a normal thing. So it's not like if you're doing it maliciously. It's like if you think about it carefully, then like you designed it to look for this, then it's okay. It's
what do you
the
mean
problem
by if you designed it to look for it? Like,
so okay, wait, let me give you the opposite example. It's like if you just ask like 100 people and ask them like thousands of questions, eventually the probability of finding certain things that correlate is more likely than finding things that are not going to correlate. So you're going to find something's going to correlate, but then it's going to end up being like shoe size and what they had for breakfast, and like those two things correlated really, do you know what I mean? So that's what harking is. So it's like if you ask enough questions and you go in and explore data afterwards, something is going to correlate. And my favorite, I actually put this in the link, it's this thing called oh, did I post it? I'll find this thing, but it's there's a link called it's basically a website that has a bunch of
the spurious correlations.
Yeah, spurious correlations. Yeah. Do you see it?
Yeah.
Can we open it
Sure.
and put it on the stream? I feel like this is a hilarious example of what happens if you do harking, because it's like you can do a bunch of, if you just mine data and only mine data and you don't do experiments, you can find correlations between the number of air traffic controllers in Montana and the popularity of the not sure if I meme. That's a bad one. I don't know what either one of those things are. Okay. So Google searches for why do I have green poop is inversely correlated with solar power generated in Bulgaria. So this is what I mean. Like you can mine data and find correlations, not causations. Like you could do it a lot.
Yeah.
Like
I will say, like, I feel like this is not necessarily just a problem with harking though. So like, because like you can also do it even with a pre-registered study where you have all your hypotheses. And I think that actually that happens a lot when kind of the way it works is kind of like, okay, if you're just like, oh, we think that there will be a gene that causes autism, and then you test like 5,000 genes to see if they're associated with autism. Like, even though you had a hypothesis, it's kind of a different problem called multiple comparisons, which basically just means that like you have you're only accepting correlations that are more basically are unlikely to be there due to chance. But if you look at a whole bunch of correlations that are individually unlikely to be due to chance, there's a good chance that you'll get one that's due to chance anyway.
Yeah. There's actually like a higher probability you'll find
Yeah.
like a fully chance thing. Do you know what I mean?
Yeah.
Like it's like a very
Yeah.
um
Especially if it's like especially if your hypothesis isn't very good. So there's this thing where it's like, okay, if the prior probability of your hypothesis being true is very low and you confirm your hypothesis, it's almost certainly because you did something wrong.
yeah.
Um,
Yeah. So the biggest thing is making your hypothesis after you've collected the data,
yeah.
and then you're just
Although I guess one of the things that this worries me a little bit, because like I see this like when people talk about this as a solution to like replicability problems, is like always have the hypothesis first. And I think the thing that worries me about that is like oftentimes we need data to like generate the hypothesis too. And I kind of
yeah.
worry about that type of research getting squeezed out if people think like kind of if you get this idea that you always need to have a hypothesis.
This is great. This is comes into the next topic, was like what causes it. And I
Mhm.
would argue that money causes it. Like money is one of the big arguments for why it causes it. So to do this correctly, you should do a pilot experiment first. And then the pilot
Mhm.
experiment is an experiment where you do a small batch and then you see a trend. And you're like, ooh, I think this is. Then using that pilot data, you should generate a hypothesis and you should generate a power analysis, which means how many subjects do I need to test for this to be true, or
Mhm.
to reject or not reject this hypothesis. Like you need to do this and then go do it, and then don't change. Like, you said you're going to collect 100 people worth of data, you only collect 100 people worth of data. You know what I mean? Like you only do
Yeah.
those things after. It's like
Mhm.
when you play pool and at the end when you have to like call the pocket and then you like do the 8 ball. Like I feel like you have to do that. You have to call the pocket and then be like, it went in the pocket or it didn't go in the pocket. Like you can't just in hindsight be like, oh wait, I called that pocket, but actually it's this pocket because it went through
Right.
that way
Yeah.
anyway. Like
So
I
it sounds
think
like you're thinking maybe the standard should be that basically like each experiment is really like two experiments.
yeah. Like people should, yeah, it's like there's two phases of it, but that's expensive. It takes time, and it is taught. I think we are taught this when when we first learned to do science in school. We are taught to do this, but immediately when we start practicing science, it goes from like pilot study to like just add more ends to the pilot and publish it and like get your job do and go. Like you don't have time, publish Paris, like that whole thing, right? Like it somehow gets muddied in the waters at some
Yeah.
point
I
along
mean,
the
and
way.
I think one thing is like I mean, I think a big part of it is like oftentimes kind of like labs just don't really have the funding to do two studies at the same scale. So like you'll do a pilot study, but that will often be more about validating that kind of like your method is actually like if you want to stimulate this kind of neuron, like the neuron actually got stimulated, but you won't really look at like the behavioral effects of that.
Yeah.
Um, I guess one question is like, okay, is it better to do that, or is it like it seems like one question is like, is it better to do that, or is it better to just have another lab try to replicate it?
Oh, Nathan,
Because
you're
I think like, you could make an argument that like, and I kind of believe in this, that having like the more data you make publicly available, the better. And
yeah.
also probably having another lab replicate it is going to give you more standardization for like, have you really documented the methods correctly?
Yeah, like that is the highest form of reproducibility. I think the first
Yeah.
form is reproducibility is you can reproduce your own results for yourself. Like I did it yesterday, I'm gonna do it again today and get the same result. That's like one level. The second level is like I'm gonna get my lab mate to do it. They're gonna do it, and then they're gonna get the same results as me. The third level is like someone else, like in a different lab, buys a similar reagent, does the experiment as I wrote in my paper, and they also get the same result. Like that is that is beautiful. Like that is the way. The problem is that no one's publishing my lab mates doing it the second time. That's not getting published. Lab friend across the country that does the exact experiment again, reproducing it, that's not getting published. You know what I mean? Like publication doesn't reward redoing the same
Replication.
yeah, like it doesn't.
Mhm.
That's the current problem, right? Like it's not rewarded, and so when money is tight, you're not going to go redo the exact same experiment someone else did before. Like even if
Yeah.
you're like like it's kind of sus, like is it real? um yeah, that's a
I
problem, I
guess
think.
like I mean, I guess kind of I wonder if that is an advantage of kind of instead of being like, okay, you do this multi-tiered thing where you redo it in your lab first. Maybe you do it in your lab, you publish it, and then like if other people think it's interesting, they're probably going to try and replicate it too. I mean, that's like where all these open science studies came from, right? And
Yeah,
so maybe that's like I don't know, maybe that's a better way to do it is like that you kind of let it get filtered a bit first and then people can find other labs will figure out like where do we really need replication when there's these like limited resources.
yeah, absolutely. I think for your, like that will make the science like have a consensus, right? Just like one person saying they got those results, kind of like, okay, whatever, they're a weirdo. But
Yeah.
like if everyone does it and they get the same thing, then it's more likely to be believed, right?
Mhm.
And that, yeah, like two people do, that's like the you know, like the silver lining for like, oh man, I got scooped. Like now I can't publish my thing in a really cool journal. And then you're like,
Like,
well,
well, at least it worked.
no, no, but then actually, then you're like sat there and you're like, wait, actually, I reproduced their results or they reproduced mine. We're all on the right track. Let's go do the next experiment, right? Like that's
Mhm.
like the, you know, the way you can, you know, talk yourself out of being like really upset.
Yeah.
uh,
Mhm.
because like this independent entity, like you almost believe it more, right? Like this independent entity, they
Mhm.
picked completely different reagents, right? Like they made their own PBS. Like I
Thing.
made my own
Yeah.
PBS. Like we did the whole differently. Their mice are not related to my mice at all. They live in different buildings 10 generations back, and they got the same result. Remarkable. Also, most likely is real,
Right.
right?
I
It's
mean,
very
that's
nice.
ideally how things should work. Yeah.
Ideally, in like a very positive, like this is how science could work.
I do
Um,
and like I do think so interestingly like so this was some of the stuff that I was reading and I think one of the interesting like sort of criticisms of these big reproducibility experiments is that kind of that when you do the reproduction attempt maybe just like it doesn't reproduce either because just the reproducers didn't run the experiment right and I think that's kind of the weaker criticism because I would argue that like okay if you're running an experiment you should document it well enough for people to like it still indicates there's something wrong. I think
yeah.
the more interesting criticism I think is that there's this interesting thing where like the more times you do an experiment, the smaller the effect size tends to get, or kind of like as you accumulate data, effect sizes generally tend to get smaller. And this kind of gets back to what we were saying about the eggs, right? So if
Yeah.
you want to reproduce a thing and you say, like, okay, these people found an effect size of X, and therefore we're going to make an experiment that is designed to detect that effect size, and we're going to use like the same number of animals there's a good chance that you're going to fail to reproduce it, not because anyone actually did anything wrong, except just that kind of that was really not enough animals to use. And it's an interesting kind of bias because it's like
Yeah.
there's publication bias. So like the samples that get published, kind of statistically speaking, are a little bit statistically underpowered just
Yeah.
because the
Oh,
ones that
and
get published are the ones where you're lucky.
yeah, I was gonna
Um,
say also your favorite. Remember that episode we did where it was like things that made you change the way you think?
mhm.
Um,
Oh yeah, the regression to the
regression to the
mean.
mean. So
Yeah.
that also happens, right? So like
Mhm.
sometimes you just stop. This is like the problem of p hacking. Like you could have just stopped when you just had enough to make the p value great, but had you gone further, like it could have gotten like you could have
Mhm.
collected too many samples and then it like it wouldn't like your p value
Right.
would no longer be relevant, right? Like so that's bad. Um, yeah, there's this like whole movement now to like straight up skip this p value business. Like it
Mhm.
is wild. But like it's good, right? Like it means scientists are thinking about it and are actively being like, wait, hold up, like is this really
Yeah.
the standard
People
we
are
should go
really
for?
thinking about what is like what should be done. Yeah.
Yeah, like, and I feel like for me that's my gut feeling, right? Like that's how I was trained. Like I had PIs that trained me where it was like it needs to be robust. Like it can't just be nearly p value of 0.05. It needs to be way better than that for us
Mhm.
to be like, yeah, we're gonna hedge our bets on this being the true thing. And sometimes it really is night and day. It's like, well, it went from like 300 to 0, and next time I got 1, and the one time after that I got 2. Like 210 versus 300 and 300 and 300 every single time. Like it's obvious. Like
Mhm.
you don't need that many samples. And like if you run a power analysis on it, they're like, you need 3 samples. And you're like, okay, cool. We're like, we did 12. Like we're still good.
Right.
But the other way to check this, and also like you're worried that you're p hacking, is if there are multiple ways to run statistics, you can do this, right? You can run the same data different ways, and generally you should publish it, for me, if it only it's statistically significant in all versions or in many versions of it. Like if it's possible, it should be that way, right? Like it shouldn't just be significant if you run it in this one specific way and no other way else, because
I mean,
that goes back to harking, right?
I mean, actually, I'm not sure if that's true, or if I'm not sure if I agree with that, because it's like all these tests have a false negative rate too. So it's like if you do, so like we talked about this multiple comparisons problem, but the opposite is true too. If you do like 15 different statistical tests, like chances are one is going to come out as not significant just by chance.
That is true, but you have to convince yourself first. Like you need to believe the data, like you need to believe what it's telling you.
Yeah.
Like
I
I
mean,
think
to me,
that's
like
the
this seems like more the case for like pre-registration. So it's like first you want to just think about like, okay, what would convince me that this is true? And then you just say like, okay, that's the analysis protocol. Like
yeah.
I think like to me that seems like a better way to eliminate the kind of harking than trying to just do like multiple different kinds of statistics on it.
I was just about to look up how many journals do pre-registration now. So pre-registration, if you remember, is like calling the pocket, right? I think that is
Mhm.
actually the way to do it. And and they did do a study. They did a meta analysis on the journals that did pre-registration, and they found that they published, like, of those papers, 60% of them had negative data that they published
Hmm.
anyway. Like so, like that's the
Mhm.
thing. If you pre-register the date, like if journals allow you to pre-register like you basically give them like the context of the study, and then like pre-register, like calling the pocket, like we hypothesize it's this, and then like you do the experiment, and then you tell them the result, right? And then if the result is negative, it's like, oh, we failed to reject this hypothesis or whatever, that or we rejected this hypothesis, then it gets published anyway, no matter what, because it was like a well-designed study and everything. And that's such a beautiful thing, because now if you do that, a lot more negative data will see the light of day, instead of being on the cutting room floor and like hidden away in the lab notebook somewhere. No one's redoing that experiment because now it's published.
Yeah. And
Like
I
they're
think
like, that's very nice.
And I think it's really interesting too, because we talk about we like this kind of gets into one of the things that I wanted to talk about, which is kind of this got identified as like a crisis, about 10 years or 15 years or so ago. And so I
Yeah.
wanted to kind of see like, okay, are any of these recommendations that people made, like pre-registration and stuff, really working? And like I actually found this study that was done. So they found 16 studies that had been done with what they called these 4 rigor enhancing practices, which were running confirmatory tests on your own data before you publish it, which is a thing that Ruby mentioned. Getting a sufficiently large sample of participants, pre-registering all the studies, committing to the methods and the hypothesis before you test them, and then make like rigorous documentation. And they found that in 86% of those studies could then be replicated by a different group. And so before, like this was what field is this in?
I think
Oh
a lot
yeah,
of
in
these are
human behavior.
cog sci,
So
yeah.
before the replication rate was around 60%, and they were able to go up to around 90% by doing that. So I think That's
That's
quite good evidence.
amazing.
Yeah.
That's so cool. Um, yeah. So I guess again, the drawback here is that now it's going to take more time and money because now there's like a journal needs to like do 2 peer reviews basically. So like they need to send calling the pocket part, designing of the experiment. That's like review number 1, and then they do the experiment. Then they like write up the experiments and results, and then they got to do peer review again, and then it can get published, right?
Yeah.
Um, uh,
Although I have seen some journals where they really don't do much peer review, like so they pretty much only peer review the registration, and then
oh, like they just
um,
do the front end peer review like really hard, and then like
exactly,
the
yeah. So
results are what they
they
are, and they're
review
like, all right, cool, you
the
did the thing.
registration, and they're like, okay, if this is a good experiment and it's rigorously described, like we're gonna publish it pretty much regardless of what the actual paper ends up being. Which is, I think they do a little bit of like checking to make sure that like doesn't have computer viruses in it and stuff, but like, but like it's pretty much they just review and part of that is also like for that thing that you said, so it reduces the bias against negative results because
Yeah.
you're committing to publish it before you have any of the data, which I think is actually also an important part of pre-registration. It's like if it's a good experiment that's worthy of being published, you should be able to tell that before the data is actually collected.
Yeah, that's it does feel kind of wild though. Like I mean in I guess in my field it is not common. Like in mouse
Mhm.
neuroscience world, I don't see that many pre-registered projects or pre-registered
Mhm.
papers.
It's
But
not super
perhaps
common here either.
yeah, it is not common, but I wonder if it would make our lives easier. Like it would make our lives like as scientists, like if we could still get jobs off of papers like this, because a lot of times what's happening now is like we do the pilot experiment, it didn't work, we trash it, we do the next thing, we try something else. Do you know what I mean? Like there's a lot of
Mhm.
times there's a lot of negative data. Like I would say 70% of the things I do don't work, and that's like a good like I feel like I'm a fairly decent scientist. um if those 70% of things that didn't work also just became publications, that could be nice. Like that would look nice
Yeah.
as good, and then you could save when you actually do make a cool big discovery, then you go for the big journals, right? So like this would be a place to put all your negative data, or even if it is positive data, you could like rewrite you could like write it up or do a follow-up study and be like, hey, we know this works in humans, let me show you, you know, more in-depth mechanism in an animal or in cells or whatever. And write up like a full, that's kind of nice. I think for
Yeah.
human studies, this is very common, much more common now.
Mhm.
Like most, a lot
Yeah.
of, almost all cog sci, people like this is like, hey, this should be our gold standard. Like they would agree that this, if you ask a cog sci person, I think they'll agree that this should be the gold standard for
Mhm.
them.
So it's
Um,
very common to do pre-registration cognitive science studies. It's not very common to do this kind of, so it's called a registered report where the journal accepts it before you have the data. Like that's still pretty rare, but it's very common to like just you create a pre-registration before you submit it to the journal.
yeah, I'm trying to think of reasons why it would not fly. Like why wouldn't they do, why shouldn't we all do it like this? And I could see scientists just like working really hard to do the pre-registration portion, like really good, write it out, and then just like not do the experiment and flag out and do you know what I mean, like not finish it.
Yeah.
Or like too many humans dropped out of the study. Like
Uh-huh.
then you have to like restart it, they run out of money, the lab shuts down, like there's so
Mhm.
many reasons like things could go wrong, and then you would just have a pre-registered thing. Maybe you have to pre-register it and not publish it, it just is pre-registered in a system, it's like you time stamped it, and then when you
Uh-huh.
do publish it however many years later, they go back to this time stamp, it's like it was like time stamped on this date. Do you know what I mean? It's like
Yeah. I
first
mean, that's
submitted.
pretty much what they do.
Yeah.
Like, yeah. But you're saying like that would count as like the publication date of the article, or
Yeah.
yeah.
I was like, could you like game the system and just sit there and like write up, like, let's say you're a new PI and you had all these ideas, you know, and you can just come up with like 20 ideas and then like stamp them all, and then take your time moseying around, like, you know, like 20 years later, come back and be like, I finally collected the data.
Yeah, so it used to actually be that you could use that, like that's how patents worked in some countries. And actually that used to be how patents work in the US, is that like if you had like a time-stamped thing that proved that like you invented this thing before anyone else, then you could get the patent for it.
Oh, it would be yours, even
Or
if
at
you
least
didn't
or
produce
at
the
least like if
prototype
someone else
until
tried to patent it, you could
uh
like invalidate that patent.
okay,
And
I could see this going sideways with people just like bum rushing all the patents during like all the versions of the experiments, and just sitting there and writing all of them out, and then like you could flood the system with it.
yeah, and that kind of happens. Like there's companies that are called patent trolls, and like their whole thing is just like they kind of like produced a bunch of random patents, and then they'll just like file lawsuits and be like, actually, we invented the nose hair remover, so like you have to
Like
pay
30
us.
years ago, here is no, but
Yeah.
like you can imagine us racing like AI. It's like, hey, this
Mhm.
AI generated 50 versions of this project, and they they pre-registered
Yeah.
all of them to this journal. So like any experiment you do, you can just like flip through the patents and be like, I did that one. Do you know what I mean?
Yeah. And I will say, like, so as of like 5 years or so ago, the US actually gave up on that system. So we now have what's called a first to file system, which actually means the person who gets the patent is like literally just the first one who gets an application approved by the patent office. And so I think it's less vulnerable to that because it takes a lot of time and money to file a patent.
Like it's an
Uh,
effort,
yeah.
effort and
Uh-huh.
time.
But it's an
Um,
interesting, yeah,
yeah, it's a like we don't have to solve, right? That's why this is
yeah.
philosophizing.
I
That's what
guess like where I was going with that is that's kind of like, at least in some systems, like that's kind of like that incentive that you were talking about.
yeah.
Um,
I mean, I would like more negative data. Like, I have negative data that I've generated that I genuinely believe other people should see besides myself. And currently, the system is whenever I go to a conference and I want to start a topic, the best way to start a topic is like, let me tell you about all the things
system that
that
didn't
didn't
work.
work for me, because it's not like an like an ego thing, but it's just like someone, if you tell someone things that didn't work for you, they're going to tell you things that didn't work for them, and it's like a
Right.
combo.
And then you
And
learn
it's yeah, and
stuff.
it's like shared information that's so valuable, and it's fun, and you make friends. It's like, oh yeah, I tried this thing, it wasted like 4 months of my life and it really didn't work. It's a good starter, but also
Social interaction tip.
yeah, yeah,
Yeah.
social interaction, but also then you're like, okay, hey, maybe I don't need to do that experiment.
Mhm.
Because I was like, oh, that's such a great experiment, beautifully designed. And they're like, yeah, it didn't work. And I'm like, okay, cool. I won't do that next time. And no, it's not published anywhere, right? Like, it's the only reason you know it didn't work is because I talked to them.
Yeah.
Um, yeah, I to this day that I
It,
feel like that is like a really good way to network, but like it doesn't have to be. It could just be published.
yeah.
I don't
And
know.
like,
It takes a lot of
I
time to
guess
publish
like,
things though. Like not to like it takes work to like make things pretty and not just like an ugly Excel graph and be like, ah yeah, that didn't work. Next. Um
I do think though, like I feel like this idea of like, okay, there's scientific journals and they kind of work with like, they work like newspapers where you make this like very polished story and then like the editor decides if they want to run it in the journals or not. Like I feel like the days of that are pretty numbered. Like I think that probably like within the next 10 years, the way things are going to start working is like you just have like an enormous mess of raw data and you feed it into an LLM, and then the LLM basically does the job of peer review and meta-analysis. And then you ask it like, hey, what genes are associated with this condition? And then it just looks through all the raw data and it aggregates things that seem to be reliable. And that's where you'll get your paper. Like
right.
we're already starting to do this, and so I think like the kind of the writing is really on the wall for like this journal system. And like basically, if we're gonna go that way, like the way it's gonna work the best is just like if we just dump all the data into a thing, and then
Yeah, and we are, I think many journals are starting that. Like this is like
yeah,
the open access like that's like their whole thing, right? It's like when you publish a paper, you literally include, here's a link, to like a not Dropbox, but like the equivalent of a cloud service that we paid for the next really long time, and you can just grab the data and you can reanalyze it yourself.
Yeah.
And it was just
But I'm
like,
thinking about even non-published data too. Like,
oh yeah, that too. And there's institutes that do that, right? Like in
mhm,
like the Allen, for example, they're straight up doing experiments. They are, doing surgeries on mice, injecting viruses, plopping them in front of a screen, and making them show them videos of things, and then you can it's literally called mouse observatory, and you can like look at the neurons doing things while this mouse is looking at a thing, and you don't it's free. Like you can just go through the experiment, and in fact, if you are like, hey, can you show them this video instead, you can write up like kind of like a grant, but like you write up a proposal, send it to the Allen, and then like it goes through like a review process, and they're like, yeah, we'll do it for you, and then they'll like do you 8 animals and
yeah.
plop them in front of whatever you want them to do, with like a nice explanation for why you did it, and then you get first dibs on that data, and then you can publish it.
Oh, neat.
Like
Yeah.
that is
Yeah.
totally a thing, like this, because like not like I love this idea because it really democratizes science. This is my like socialism science thing. Like
Mhm.
people from countries, or future US when we have no money, anyone with a computer could take this data, like from like high school. Students from anywhere could just sit there with this data and play with it and find things. And what a beautiful way to teach kids like how to do science. Like
Okay.
they can
Yeah.
actually work with data like without it costing an arm and a leg and mice and time and reagents and stuff. They could just have the back end of the data, like get them into it. And it's like data with like a safety net. Like the worst thing they could do is waste their time.
Mhm. Yeah.
Like the positive is that they could make discoveries, and um like we could just forward science that way. I don't know,
Yeah.
positive
And yeah, I
vibes.
think like there is that, and I also wonder like, can you do it like even with less like the Allen system is a very centralized lab that's gathering all this data. But we're also really interested in like, could you do it in a decentralized way? And I'm thinking about it's like there is this one technique for improving language learning and sleep, and it didn't work. Except it worked really well at this one lab, and we're pretty sure that the reason is because they played their sound cues more softly, and so everyone else is disrupting people's sleep, but this particular lab wasn't. And so basically the people who are getting negative results weren't publishing them. But if that data was available, and especially if the raw data was available, you could actually do an analysis where you, instead of just being like, maybe this works and maybe it doesn't, you could actually like say like, huh, what factors predict if this works? And you can basically use the attempts at it at different labs that have been done as sort of different conditions so that
Yeah,
you can see what works. And then
yeah.
that's just like a thing that you can do if everyone just kind of like dumps their unpublishable manuscripts and data
Yeah.
somewhere.
No, I was gonna say that is kind of the beauty of something that is not reproduced. So like I did the same
Mhm.
thing, and then like I thought was the same thing, and I got a different result. And then being able to compare what are the actual things that you did differently. It's like, well, I used this speaker, right? Like this brand of
Yeah.
speaker. And they're like, we use this brand of speaker because why would you think it's any different? And then you could find, you can find like, hey, this one has like a weird vibration at the end, and then you're like, wait, is that vibration the sound that actually works for blank? These are opportunities to make science discoveries, right? Like it's
Right.
not just like a
Mhm.
negative. Like I think we started the thing and be like, oh my god, a reproduction crisis, like science is a real.
Yeah.
But here is like, here's an opportunity that we're kind of wasting by not
Yeah.
not sharing.
Like, yeah, it's
It's a
like, I
cultural
guess,
thing, right?
yeah, like that's interesting. Like the other way of thinking about this like lack of reproducibility is like it's, hey, free data, because now we have data on like when this works and when it doesn't and what factors make it work or not.
Yeah.
Like
Yeah, we
like it's
are
like,
working against the culture of like, our join our lab if you want to use this data, and you know, like that's why you come here, right? Like it's like the opposite of that. It's like doesn't matter where you come from. It has like you're working against, people who have been in the system for a long time. It's working for them and they don't want to change it.
yeah. But also that kind of analysis doesn't work unless everyone pools their data. Like
Yeah.
it's not like that you're kind of anyone's losing out on something. It's like it's a thing that you fundamentally couldn't do unless people cooperate.
Yeah. And
um,
that's kind of the thing. Like when you to generate this data, it's really expensive. It's expensive to house animals, it's expensive to collect them. Um, like I understand people's hesitancy to just be like, here's free data for the world so you can scoop us with it. I can see the hesitancy for it, but I can see so much growth for it too. Like I can see the double-edged sword. I see why people could be scared, but on the other hand, maybe my mind would be changed when it's like my money that was used to generate the data and not my PI's money. But from my perspective, this is forwarding science, and we should
yeah.
be chasing the opportunities to to make discoveries faster. Um, yeah.
Honestly, in my experience, the biggest barrier is not that like people have philosophical objections to it. It's that like formatting the data to get it into the data repository is really annoying.
I was gonna say, our lab recently started this thing so that we wanted to put everything on Dandy. Um,
Uh-huh.
uh, like, and we even in the lab ourselves, just with people that we work with and respect and like have no beef with, we have trouble agreeing on like how the format the folder names.
Yeah.
Like we were like, no, we need to name every folder and it needs to follow each. Like there's so many different ways to save the files, and it not just differs between lab to lab, it differs person to person. And then now you want to try, do you know what I mean? Like just to get this to work, like we all have to agree on a system, and sometimes there is no perfect system. And
Right.
yeah, it's hard. And it,
Mhm.
you know, you could spend the time sat there reformatting your data for like 6 months, and um or reformatting someone else's data so you can understand it like with your code, like that's rough. um
Yeah. Because I feel like a lot of these formats and like a lot of the software and data formats are really purpose built for a specific experiment too. It's like, okay, this is what makes it easiest to do the experiment.
Yeah. So our lab has been recently trying to get all our data Dandy. From Dandy stands for, actually I have no idea what Dandy stands for. Dandy, Brain Initiative Supportive Data Archive for Publishing. No, that doesn't know. Distributed Archives for Neurophysiology Data Integration. That's what it stands for. That's what we're trying to do so that all future publications we can just like put in the raw data so that anyone can look at and analyze our data as well, which is kind of nice. Like I feel like there's still a gap between wow, that's so nice theoretically, and here is it implemented. Is going to take six
Yeah.
months for a postdoc to sit there and do nothing but this.
Yeah. Uh-huh.
And we're like, he could be doing experiments or she could be doing this. Like, do we want to waste that time?
Right. Yeah. Yeah. I guess
Money.
one that kind of reminds me of another thing that I wanted to mention, which is about kind of the organizational part of this reproducibility stuff, which is like, so I think when you started talking about it, so like we mentioned, like it kind of when this first came out, it was recognized as like a problem in psychology. And like oftentimes when people talk about it, it actually like I think people often really talk about it as like a problem that is in social science or human behavior or human biomedical fields specifically. And I thought it was really interesting. So um, Nature did this survey back in 2015, which kind of gets at this. I thought it was super cool. So they pulled 1,500 people, um, and they looked at it by field. So they asked people like, okay, how much of the work do you think in your field do you think is reproducible? And so what they found was that chemists and physics and engineering people were like most confident in their published work. So they said like 70, 80% of the work they thought was reproducible versus like medicine and other fields. So like sociology, psychology, this was like maybe 50%. So so they were very confident in it. But then when you looked at kind of when you ask people like, have you failed to reproduce an experiment? Actually, like chemists failed to reproduce the most experiments of any field. And so I thought this was really interesting because it suggests that like there's still some like field specific things going on with
No,
this.
but
Like,
fail to reproduce your own experiment is also quite high at
yeah.
above
I mean,
50%.
it's high for everyone. It looks like,
Yeah,
but yeah.
wild.
Um, and of course, like, I mean, you could say like, okay, maybe it's like chemists and biologists just tried to do more reproductions than other fields. But I thought it was really interesting how kind of it seems like there's these fields where people are like, yeah, our field is super reproducible, and then like 80% of the time they're trying to reproduce experiments, it's not working.
Yeah. That is a little scary.
And it's like, and like, in addition to being scary, I think it's interesting because I think we're both like in neuroscience, like this is a field where this is like talked about a whole lot. But actually, like, I mean, I'm in an engineering lab, like right now, and I think one of the things I notice is like people like, I think I am only aware of one case where someone tried to reproduce an experiment ever in the whole lab and like it didn't work. But like at the same time, like we don't really talk about it at the to the degree to which like biologists and like neuroscientists and stuff are talking about it. Like it seems like this field just hasn't really caught on to that yet.
Yeah. Yeah, it varies in different fields. I wonder if um like there will be a reproducibility crisis in your field in 5 years or 10 years. Do you know what I mean? Like we like our reproducibility crisis happened 10 years ago. Like it started to become like this thing that everyone talked about, and we were taught to think about it carefully, like really think about experiments and like aggressively try not to be hack. Like this is the
Mhm.
like it was like a thing that it was just drilled into us, like this is how you do it, this is an ethical way to design experiments, this is how you I wonder if that's going to happen in other fields at a different time than ours. Do you know what I mean? Like it's just happening, and I think it will happen more when there's less stigma, because right now it feels like, hey, you're lying, and it sounds like you're doing something. Do you know
Yeah.
what I mean? When you're like, you fit this, your data is not reproducible. Like I'm scared of the boogeyman, right? Like I'm scared if someone else can't do it, but like I am happy to talk to them. Like there have been instances where people are like, hey, for your experiment for that paper, did you do this? And then they would have emailed me, and I was just like, oh no, you have to use this particular tubing. Like
Yeah.
like it doesn't have bubbles, and if you have bubbles it'll be bad. And they're like, oh okay, that makes so much sense, like I'll go get that bubble, that bubble free tubing. And those are the kinds of like tips and tricks that we should be willing to share at any time, because it would
Right.
allow them to not no allow them to reproduce your data. Do you know what I mean? Like
Yeah.
they're
So like in these fields, it's not stigmatized, is what you're saying. Like,
I think it's not for, I mean, yet, or at least there's less
mhm,
toxic people in our field now.
yeah.
Maybe I'm just, it's like I feel really good about the people in my field, and like a lot of them are very nice humans and generally are there for good reasons. So I'm like, like I'll tell you how I did it specifically. Like I didn't use this. I used this kind of glue, and
Yeah.
that glue sucks. Like we should share these things openly,
Right.
and it will generally make science better. So why not?
Yeah. And I feel like similarly, like I feel like in the aspects of neuroscience, like at least with most of the people I work with, it's less like there's now less this idea of like, oh, you're trying to prove me wrong. And people are more just curious. Like people are more
Yeah.
just like, what if the volume does matter? That's super interesting.
Yeah, like
Like,
exactly that matters of like the friction of,
yeah.
the material that you use.
Uh-huh.
And the other thing is like the stigma behind retractions. I feel like this is a hot topic. I don't know, like I've been thinking about it a lot. So before the stream I was looking up like retraction rotch and like
Hmm.
like how long it's been running. It's been running since like around 2015 maybe. It
Hmm.
was definitely running in 2015. And I remember it was kind of a big deal because it was kind of scouring all kinds of journals and all kinds of places, and I was looking for retractions. And I was like, wow, this is like the T, like this is the T site. Like you could look for things that
Yeah.
went wrong. But actually the first paper on retraction watch is like from like 1800s or something, and it was a self retraction. and, and it was like the person being like, oh actually I I wrote this at the time, but now I think it's not true. And the ability to do that without being it without the stigma, I think that is something that we need to normalize. Like we need to bring that back. Like we should be able to publish something like confidently when we believe it as it is, but also know that hey, if you followed up with a bunch of studies and other experiments and you show that actually now we think it's another way, it should be okay for you to just in the future publish a thing that says actually I was wrong last time. This is actually correct. This is more, do you know what I mean, without it
Mhm.
being like oh my gosh this person's wishy-washy. And I was like no, like at the time with the data they had, that's how they interpreted it. In the future they did a new experiment, they did like 20 more replicates of it, and they're like okay actually no, never mind, it really is a different thing. And it should be okay for them to say ha, I was wrong last time, oops, like now I'm right.
Right.
And not have it detract from their um standing as a scientist, right? Like
Mhm.
like I'm just thinking about like future if the boogeyman gets me and I was wrong. It's like how would I like it to be? And I was just like, well, if I was actually wrong and I published this thing, 20 years ago during my PhD, then maybe I, here's a new proposal for what it could be, and do you know what I mean? Like, and not be um negatively like the public wouldn't have a negative output or negative viewpoint of what I had done. Anyway,
I
okay,
do
maybe
think
this was just the boogeyman anxiety that made me want to do the stream.
like I mean, I do think that like it's not exactly about retractions, but I do think that kind of like I don't think scientists get judged that much for like making mistakes. Like I'm thinking about like Linus Pauling and vitamin C. Like Linus Pauling is a famous chemist, but he also thought that like taking like enormous amounts of vitamin C would treat like different diseases. And he was actually like he never really gave up on it. But people are just like, okay, you were wrong about some stuff. Like whatever.
Yeah. Um I was thinking about other ways to like make sure, other ways to improve ourselves, like self-improvement to
Mhm.
make the reproducibility crisis like not or like at least not add to it. And
Yeah.
one of the things that I really enjoy or feel safe about doing is implementing double blinding.
Hmm.
So a lot of
Mhm.
times you can do single blinds, but I really like double blinding.
Mhm.
Like I want neither me nor the person doing the experiment to know which subject is the control and which one is the actual test animals um until all the results are in. And actually
Mhm.
not just once the results are in, analyzed and plotted, and I run stats on it and just group A, group B, and then
Right. Yeah.
afterwards I unveil it. And I know that sounds like really controversial and really like day to day it actually just means like I assign colors and letters to a lot of things, and then I
Right.
have a folder that I tape or like a index card that's taped to the corner of my desk, and then I don't reveal which one is which until we finish
Like
the
you just get a
experiment.
student and you're just like, hey, could you decide if like the active group is blue or red and then put it
Correct.
in this folder?
Yeah, and put it in
And
the
then,
folder, and then like they're not even related to experiment. They're
yeah.
straight up some random person in the lab, and it's just sealed, and it's sealed there until I finish the analysis, and then I'm just like, oh my gosh, it's so exciting, but also it could be not exciting. Let's see. Um, I, I, it
And it's
feels
interesting
safe.
because it's like we agree that like that's how you should do it for clinical trials. Like it's kind of surprising that it's not more common in other
Yeah.
aspects of
Yeah,
science.
I basically don't believe in behavior experiments that are not blinded anymore. Because I'm just like,
Mhm.
you can want an animal to think that. And they're like, it doesn't matter if you use a timer, it doesn't matter anything. You could be like, I'll discount this animal because he was like scratching his butt and like he wasn't paying attention. Like, you can have biases and you can apply it and you can be like, oh, like, if they eat their poo during this time, we're not going to count it because they're not paying attention to the stimulus. I'm like, well, no, like, we're going to be completely
Yeah.
agnostic to everything and just count it as it is. And it's like, well, it fell off in 2 seconds. Like, doesn't matter if he's eating his poo, doesn't matter if he like was licking anything, doesn't matter. If it's blind,
I
you
mean,
just
yeah.
yeah.
I mean, there's also like, have you heard of like the clever Hans effect?
Oh yeah, that's
Yeah.
that's one of the boogeymen that we can fight. So that's this horse that can do multiplication or something, right?
Yeah, well, like nominally, but I think like the point in this context is like both animals and people like the way that you interact with them when you're running the experiment can also influence their behavior. Like I'm doing an experiment where we're trying to actually see if we can give caffeine transdermally. And one of the reasons we're very careful about double blinding it is because we know that kind of like if the experimenter seems like more tense or is behaving differently in any way, even unconsciously, that will actually influence the subject's physiology. So we want to like make sure that that isn't a confound for this
Yeah, and if
experiment.
the patient knows what they're supposed to display, or the
Mhm,
subjects know what they want to display, they have humans just have this thing where they want to please people. And so they will try and guess what would be like a more optimal, so like the performance, and they will
mhm.
do it. And then that's what happened to the horse, right? Like
Yep.
the
Uh-huh.
humans started nodding and smiling when when the horse got, to the correct number. And so like the animals seemed like they could count or multiply or whatever, because the experimenter was so excited. They were like, yeah, 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 okay, good. And then,
Yeah.
um
And that like probably happens in a lot of experiments. Like there's probably a lot of experiments. It's like, oh, we found some intervention where does this thing, and it's like it was like really because like the experimenter was like staring at the rat training box, being like, "Go rat, go!" And the
yeah,
rat was like getting stressed out and like.
yeah. Yeah, or you're in a room, you're stressing them, they feel
Mhm.
your cortisol, who knows? Like any of those things. It's just good practice, and that's something we can do and control. And when you get a result that is real, there's no doubt. Like, right? It'll definitely
Yeah.
you did it. You did it right. I like that. Sometimes like the first pilot I might not do it. But like any data that like I eventually will plot or put in the paper or whatever, I do it all blind. Which is like the best way to do it, and it feels safe. It feels good. If you can do it, do it. I was trying to think of other things that could be really useful. um oh, here is like find a buddy to do it. Like just do one of your trials. So this was really useful when I had a student. So like I would do part of the analysis and I'll show them how to do it, and they'll do the majority of the analysis, and then we'll go and cross check how we've done the analysis with each other. Because we'll just be making, there's a lot of times where there's judgment calls. It's like, hey, did you call this a cell, or you call this one not a cell, or this one debris? And we'll go and like cross check each other. And then by the time the date, like the full set of data came through, we had figured out a system where we both called similar cells. Like we had like a 90 plus overlap in during the judgment calls, which is really nice. And so if you have a buddy to do it with, um
You can kind of cross check.
yeah, you can cross check and you can do more, right? Because there's only like one of you, which is very nice. You got to do the pocket first, the pre-registration style thing. And I think in practice, if journals don't let you do that, I think a lot of times we have to write things like grants and proposals and stuff. It's kind of nice to lay it out then, and then you can use it later in the actual papers. But it's also like nice to like clean your thoughts and then have it sat out, and so that when you do the experiment, um, you can adjust as needed. Learn stats.
Yeah.
um, oh, mine was write better in methods. I think my pet peeve, I think it's a lot of peeves for a lot of scientists, is going into papers and then finding the method section. It was like, as done in previous paper citation. And then you go to that citation paper
And
and
then
then
it's
like,
like
as
impossible
done
to
in
find
previous
where the method actually is.
citation. Yeah. And then you're like 7 papers deep. And then that paper is like not even digital anymore. Like you have to go to the library and find it. I have done this before, and I was just like, are
Yeah.
you kidding
Same.
me? Can someone just write it down one time? And I was just like, wait, I should do that. And so okay, so I think I've adjusted my series my expectations for myself from being like rewriting it every single time. I was like, okay, in your tenure in a lab, so in the time you are in that one lab, you can refer to a paper in that same lab while you were there. Like as a grad student,
Hmm.
like if you
Mhm.
published a paper in year 2, you can refer to it in year 5. But once you've moved on to a different lab, you need to rewrite you need to rewrite it again, because you're going to use slightly different reagents, slightly different things in that amount of time. Like I feel like the time window for me is like no
It's like
more than,
while you're in the lab.
yeah,
Yeah.
while you're in the lab. So like less than 10 years, like you can be like, yes, refer to the paper before. Please do not refer to the paper before for paper that is not in your lab
I'm just like
and like
I'm
someone
just
else's
like
lab.
put the methods for the study in the methods section. Like honestly, like it's not like there's really a space limit. Like
Yeah.
just like it makes everyone's lives so much easier.
Yeah, actually,
Like
there is no space limit. You're right.
it's like
Like the text has
that's
space limit, but methods
yeah,
don't.
like this used to be a thing, and like I so actually the reason this is more common for older papers is like it used to be that like journals would limit how long the methods section is, but that's done like much less
Because they
frequently
were
now.
printed
Yeah,
on paper with ink
yeah.
and with
Mhm.
killed trees. They're like, yeah,
And
every line counts.
yeah, every line costs us money. So, but yeah, now I'm just like, okay, like the point of the methods section is like that you put the methods in it. Like, like, yeah. Um, speaking of methods and statistics, though, I think like one thing that I was thinking about, like, because we were, so this is the thing we wanted to talk about is like, oh, how like what might we do to improve this problem? And I think one of the things that we actually should be doing is really improving and standardizing um, how we teach statistics and experimental design. Because like I thought that this was like a thing that pretty much everyone learned until like I joined an engineering lab, and like I realized like actually almost no one in this lab like actually takes anything about statistics and experimental design, and like everyone has to kind of learn it on the fly, and it really doesn't work well.
Yeah.
um
You shouldn't be having to learn statistics while you're writing a paper to be published. Like
yeah,
this should
yeah,
it should be like
uh-huh.
in I don't know, maybe this is like a they should learn in undergrad. Like it
Like
should be
or
like
in grad
intro
school, I mean, so
bio.
these are
Yeah,
grad students, like there should just be a
sure.
requirement that
Yeah,
you
yeah,
take
like
that
stats
statistics
for grad
course,
school. Yeah.
which like my
We actually
neuroscience program did
mine
that.
did. Yeah,
Yeah,
my program did.
yeah.
Um
But
maybe the
in these like
engineering ones need it.
engineering programs, there isn't that really.
Please teach them experimental stats.
Yeah, I mean, I'm trying. Like, I explain to all the students about like why they'll be like, do we really need a control group? And I'm like, yes.
You'd be like, this is not publishable without any
Yep.
control.
But like, I think the important part is like having a standardized way where it's just like, okay, everyone who comes into the lab needs to know this, because like that's how you like really get like a lab culture of doing good rigorous research.
Yeah, I was
Also,
actually. Oh, go ahead.
I was just gonna say I'm gonna continue roasting engineers, but like the other thing that we do that I find really weird and like really not good is, so like in some fields actually a lot of the papers you publish are conference papers. So you submit them, like you submit the paper to the conference and then it's published online, but like if you it gets accepted, you go and present it at the conference too. And like the problem with these that I've seen is like they all have deadlines. So you have to submit it by a certain time. And like, the thing I noticed is like deadlines really make people cut corners on rigor. Um, like there's a lot of cases where people are like, oh yeah, we really should do it this way, but we don't have time to. Um, and so I really think we should just like get rid of deadlines for stuff. Like I think all papers like should just be the paper goes in when it's done. Like, and this kind of gets back to your replicating, like, oh, running internal replication studies too. Like no one's gonna do that if you have these aggressive deadlines. Like
Yeah.
when with these conferences, what happens is people do all their analysis like the week before the conference, just because that's like naturally what people do. So
Yeah.
I think, yeah, get rid of
I was
deadlines
gonna
for
say,
stuff.
oh, that's such a rough thing because if you don't get rid, if you get rid of deadlines, I don't know if I would get anything done. I feel like I'm motivated by like I
But like
put
you
red
submit things
things in my
mostly to
calendar.
journals that don't have deadlines, right?
Yeah, that's true. Most journals don't have deadlines. You have personal deadlines. It's kind of like I really want to get this paper out before, vacation or before someone else gets their paper out.
Yeah, I
Like
mean,
that's
I
the
do that
general
too.
like the two
I feel like
motivators.
I feel like that is more okay because I also feel like with those, I can say to like the people I'm working with, like, hey, I actually don't totally understand what's going on in this part. Like, we need more time. Like,
Yeah.
or like,
Yeah.
let's talk to a statistician and make sure we did this correctly.
Yeah. I also I had a really good experience with this, and I wish it was more common. Which is where if you find out a different lab is doing a really similar project and working on a similar question, you guys kind of just get no, we just like clock each other and be like, hey, how far are you? How far are you? And then it's like, can you wait for me? Or I'm only a
Mhm.
couple of months off. Like, let's publish together, and let's, and then like it actually makes the stories better, especially if they conflict or in conflict in like minor ways. Like that's a discussion for like why is it different? Like that's how you make discoveries, right? Like I said, like it's a replication. um that's such a great way to do science. Imagine if we all did that all the time. And then, do you know what I mean, in science, it'll be really, I think it will be really good for the field in general.
Mhm. Like if
To
there was some sort of like clearinghouse where people could be like, oh, what are you working on?
Yeah, and I think it requires trust, right? It needs
Yeah.
your, like it worked out for in my field and with the people I worked with because they trusted each other, and like they didn't want to burn any bridges, and they're like, hey, like next time we something like this happens, let's do that thing again. And it works out really nice. Or even like let's straight up collaborate, right? Because like it was like the beginning of a really beautiful collaboration, and I think that's really nice. I imagine that it wouldn't work with everyone, but if it could happen, it's like a really beautiful way to do science.
Yeah. Yeah.
um yeah, and we should do it as much as we can.
Mhm.
With that, actually, we are over time. Thank you so much for joining us. We are Philosophising. Until next time.
Yeah, that one ended on a positive note.
Yeah, positive notes. We tried to
Yeah.
be positive. I think
Uh-huh.
I
But like
was
the
really worried
last
about
stream
it
we
being
really
really negative.
failed.
Was it at being positive?
I think like the last stream we ended up talking about like deadly genetic diseases or something
Oh,
at the end.
yeah, that's true.
I forget what it
Sorry.
was. It was really depressing, but this time it worked.
Yeah, yeah, positive things.